Thank you so much for the opportunity to present our work today. My name is Madalena. I'm a postdoctoral fellow in the lab of Alex Shearer at the Biozentrum Basel. And I'm really interested in understanding how translational control shapes gene expression as embryos develop. So upon fertilization, transcription is silent. So embryos rely on maternal products for early embryogenesis to take place. This is termed the maternal to zygotic transition, MZT, where maternal control is handed over to the zygote upon zygotic genome activation. And I study this developmental process that is conserved in the zebrafish embryo, which within 10 hours goes from a single fertilized embryo to, or a single fertilized cell to an embryo with more than 25 different cell types. And we've learned in the past decades a lot about the transcriptional and post-transcriptional mechanisms that regulate mRNA abundance changes throughout these dynamic transitions. But it's also known that often mRNA levels are poor predictors of protein abundance. So the question that I was interested in understanding is, how do embryos orchestrate mRNA translation during these dynamic transitions? Or in other words, how do they know how much protein to make and when? So if you look at translation from the perspective of an mRNA, there are several features that control translational output. But for translation to first start, the small ribosomal subunit must first engage with the 5' cap and unidirectionally scan the 5' untranslated region, or UTR, until the ribosome engages with the first AUG for protein synthesis to take place. So the initiation is a major rate-limiting step for protein synthesis and also a possible point of control for selective mRNA translation. And we've learned mostly from yeast and cell studies of features in this 5' UTR that can impact translation, such as hairpins, upstream open reading frames, or the Kozak sequence. But I think we lack a systematic understanding of the contribution of these 5' UTRs to translation regulation, how do they cooperate, and also which additional features that are yet unknown that could contribute to regulation. So in my postdoctoral project, later in collaboration also with the lab of Georg Seelig at the University of Washington, I wanted to understand if the 5' UTRs can impact translational dynamics as embryos develop, and also to uncover features that may regulate protein synthesis. And it has remained challenging to understand the contribution of 5' UTRs to translation control for two main reasons. One is because other features like the coding sequence and the 3' UTR also cooperate to translation. And the other is because usually 5' UTRs are not well annotated, which is why I first actually recovered or re-annotated 5' UTR sequences of endogenous transcripts that are expressed throughout early embryogenesis. And then I employed a reductionist approach where we put these endogenous 5' UTRs in the context of a reporter so that all of the other features are the same. And this gave rise to a group of 18,000 unique 5' UTR reporters that also have different lengths of the 5' UTR that we inject all at once into the fertilized embryo to employ a massively parallel reporter assay, or MPRA, throughout early stages of development until the end of gastrulation at the bud stage. I then perform polysome profiling, which is a biochemical approach that allows to separate these reporters based on the number of ribosomes that are bound to this superfold GFP CDS. So if there's a 5' UTR that leads to poor ribosome recruitment, these are going to be physically in the ADS fraction of this polysomal fraction. But if there are enhancing features in the 5' UTRs, this is going to lead to enhanced ribosome recruitment. And these reporters are going to be shifted towards the right on these polysome fractions, towards the low molecular weight or high molecular weight fractions, which is a proxy for enhanced translation. And by performing high throughput sequencing to recover 5' UTR reporter information, we can then couple 5' UTR sequence to translation initiation behavior. And we analyze this data by then calculating a ribosome recruitment score, or RRS, which is the ratio of ribosome bound to total available for translation reporter at each stage of development. And this heat map shows you a global overview of translation initiation dynamics, where each row indicates a different 5' UTR reporter that was recovered throughout the entirety of the developmental process, and each column represents one of the fractions at each of the stages. And you can see that 5' UTR seem to be sufficient to confer different initiation dynamics. And we also wanted to make sure that when we calculate RRS, this actually serves as a proxy for protein synthesis capability. So we took 3 reporters that lead to high, intermediate, or poor ribosome recruitment throughout the development, and we injected them each separately with a dextran dye control. And for example, on the left panel, this is the 5' UTR that leads to low ribosome recruitment, and you can see that there's almost no superfolded GFP expression, whereas the ones on the right, the 5' UTR of the HNRNPL, which leads to high ribosome recruitment, leads to already superfolded GFP production at 2 hours, and this is greatly enhanced throughout development. So this indicates that RRS is actually a good proxy for protein synthesis output, and that at least in this developmental context, initiation seems to be rate limiting for protein synthesis. So what did we find? We first looked at features that had been shown to regulate initiation, and we focused at the 10 hours post-fertilization time point for this. So here, what I'm showing you is that we recapitulate the generally inhibitory impact of UTRs on downstream translation. So these violin plots represent RRS at 10 hours post-fertilization, and you can see that the higher the number of predicted UTRs, the lower recruitment you get. We also looked at the translation initiation site sequence, or the TIS, and we find amongst the top 10% 5' UTR recruiting reporters the sequence AAC-AUG, which represents the Kozak sequence for the zebrafish, whereas the bottom 10% have no clear enrichment in sequence. So this means that we can recapitulate biologically meaningful information, but also recover sequence information. And we also found that in vivo, the 5' UTR length is very important for regulation of initiation. So the longer the 5' UTR length, the less recruitment you have. But what about other elements, such as motifs? And this was really the motivation behind this assay for me. So we took a simple approach at first. We looked at the quantile data, so the 5' UTRs that are most enhancing or most repressive across development, as you can see on this heat map. And we also performed soft clustering analysis to identify groups of 5' UTRs that have concordant behaviors as embryos develop. So for example, if you focus on the two bottom rows of this heat map, you can see that there's groups of reporters that seem to lead to an increase in translation initiation until gastrulation sets in, and these become repressive at the end of gastrulation. And then we perform motif enrichment analysis to try and identify motifs that might be enriched in these different groups. So this is a schematic representation of the behaviors that we observed. And we were really excited to find 86 motifs that are enriched differentially between these groups that we termed enhancing, dynamic, or repressive based on the behavior that is associated. And my current favorite hypothesis is that these short sequence motifs are actually recruiting RNA binding proteins, or RBPs, in a sequence-specific manner to regulate translation. This is in part because RNA binding proteins and the motifs that they bind to are highly conserved. So here you can see the binding motif of LF-like family of proteins that are conserved from C. elegans to humans, and they recognize this specific sequence UUUG, UUU. So I was very excited to find amongst the dynamic motifs this same element. And we also find other examples of sequences that have been shown in other species to recruit a specific RNA binding protein. But in most cases, we actually don't know what that protein is doing when it binds the 5' UTR. And even most excitingly, about half of the motifs are non-characterized, meaning that at the moment, we do not know if and which RBP they might be recruiting and what the regulatory outcome is. And finally, I'd like to leave you with the idea that 5' UTR heterogeneity might add an additional layer to translational control during these dynamic transitions of the MCT. So for some of the 5' UTRs, or for some of the transcripts, there's actually a 5' UTR switch that occurs depending on if the mRNA is deposited from the mother or zygotically expressed de novo. So these are transcripts that bear the same CDS, so they give rise to the same protein, but they have a different 5' UTR. And we included these in our assay. So this is an example of two 5' UTR isoforms of the same transcript that leads to differential ribosome recruitment. And when we actually look at the sequence composition, we observe that the maternal longer 5' UTR actually has 3 motifs that match. So we postulate that heterogeneity or switching of these 5' UTRs changes the length, UORF number, and also motif grammar to regulate translation initiation dynamics. And finally, in a collaboration with the lab of Georg Silig at the University of Washington, and specifically Sebastian, a postdoc in the lab, we use this data to train a deep learning model called Dania 5', which is a quantitative model of 5' UTR mediated regulation. So based on the sequence of the 5' UTR alone, we can predict ribosome load and also motif contribution importance. So here, to showcase the power of this model, I'm just showing you two different 5' UTR sequences that have different lengths and also different sequence composition, including two different scoring motifs. So if the model predicts their behavior based on the length alone, they are different as expected because length is important. But when we consider the sequence composition, then the predicted behavior is different, and it actually matches quite well what we measured in vivo. And even more excitingly, when we shuffle these sequences to disrupt motif but maintain nucleotide composition, we see that the behaviors no longer match the observed measured behaviors. So this to us suggests that the motif grammar is important for regulation. So in summary, I've shown you that we've developed this in vivo MPRA that reveals that 5' UTRs are sufficient to confer translational dynamics during embryogenesis, and that this allowed us to identify more than 80 motifs with putative cis-regulatory roles in translation. Currently, we believe that these motifs might be recruiting RNA-binding proteins in a sequence-specific manner to regulate translation dynamics, and perhaps that these 5' UTR switches might be a means to control cell type-specific regulation. And this is, of course, what I'd like to explore in the future. So I really like to determine motif causality and identify motifs that are required and sufficient for regulation, identify the RBPs that might be minding these 5' UTR motifs, and of course, explore their developmental roles in the embryo. So with this, I come to my acknowledgements. I'm really grateful to Alex for providing me the freedom and opportunity to explore my interests, the collaborators that I already acknowledged in the presentation, and other collaborators from the work from my postdoc that I invite you to read. Support and funding, again, the opportunity to present here today, and I'm happy to take questions, and I thank you for your time and your attention.